Welcome to my channel and welcome to St. Joseph's College. Welcome back and welcome to a new video on culturing microbes. A microbial culture is a method of multiplying microbial organisms by letting them reproduce or multiply in a nutrient medium under artificial laboratory conditions. These are the basic instruments that you require in a microbiology lab. You require a spirit lamp, and an autoclave is required for preparing the medium. You require an incubator to grow bacteria under particular temperature. You require an oven. You require laminar airflow or inoculation chamber for transferring the culture from one to the other. And most importantly, you require an inoculation loop, and with the help of this inoculation loop only, all the transfers are done under laminar airflow. This is the outline of the preparation of the nutrient medium. You weigh the exact amount of nutrients based on the literature, based on the recipe. Then you mix it in water, maybe either adding agar or not adding agar, depending on whether you want to prepare a liquid medium or a solid medium. For a solid medium, you have to add agar, a gelling agent. For a liquid medium, no need of agar. So after preparing it, it has to be sterilized by using an autoclave, that is stage number 3, step number 3. Then you are adjusting the pH in step number 4, and in step number 5 you are dispensing the medium onto the containers, maybe petri dish or a test tube, and then you are storing them, and later you are using these media for inoculation. There are different types of cultures. Basically we have pure culture, subculture, mixed culture and contaminated culture. A pure culture is a laboratory culture that contains just one species of organism, and microbes are usually found in mixed cultures. And in pure culture, only one particular organism is growing, and this was developed by Joseph Lister in 1878 by serial dilution. A pure culture is usually derived from a mixed culture by transferring a small sample into a sterile growth medium. And there are different methods of assessing the purity of the pure culture. And the second type of culture is called subculture. Is a procedure of transferring the microorganism into fresh nutrient medium from its stock culture. It includes transfer of culture from slant to slant, slant to plate, plate to plate, plate to slant, solid medium to broth, or broth to solid medium. Subculturing allows an analyst, a scientist, to move microbes from one set of parameters such as temperature, media to another. This information is useful in microbial identification, as some species will grow and some will not grow depending on the parameters chosen by the scientists. This is what is happening. This is what you're seeing subculturing on the screen here. You have a old stock culture, and from there, by using an inoculation loop, you are transferring onto a fresh medium onto a different slant. Culturing of bacteria. There are different types of pure cultures. You have pour plate technique, streak culture, stab culture, stalk culture, starter culture and enrichment culture. So these are the different types of cultures. We will see some of the important types of cultures in this video. This is what is collection of bacteria from the stock culture by using an inoculation loop. You can see the inoculation loop is being used to transfer the cultures onto a different medium. You are seeing the streaking happening here. This is called streak culture. Take the microbial cultures by using an inoculation loop and run it on the nutrient medium in the form of streaks. you stick it onto the nutrient medium, as you can see it on the screen. This is called streak culture. So coming to the next type of culture called pour plate culture, in which what you are doing, you are pouring the sample onto a nutrient medium so that uniform distribution of the sample or inoculum occurs onto the fresh nutrient medium. In step 1, you are collecting the bacterial sample. In step 2, you are pouring the sample onto a sterile nutrient medium. In step 3, you are swirling, mixing the sample, that is the inoculum, that is the bacterial culture, onto a nutrient medium so that there is uniform distribution. In the 4th step, you can see the bacterial growth occurring in the form of colonies. You can see here how pour plate technique is used, and you can also see how the bacteria grow as colonies, and the colonies of different types of bacteria are being grown. It's a mixed culture. The next one is called slant culture, where the nutrient medium is available in the form of a slant. It is not flat, it is slant. A slope kind of media is available so that you have a larger space, larger surface area, and that is called slant culture. A stab culture is one where inoculum is taken with the help of an inoculation loop, and the inoculation loop is stabbed into the solid medium. It is pierced into the solid medium, whereas by which you are introducing the cultures deep into the medium. So you are seeing on the right hand side the stab culture, where the microbes are stabbed. They are inserted deep into the nutrient medium. That's called stab culture. One is slant culture, the other one is called stab culture. So you can see the picture showing stab culture, where the inoculation loop is stabbed along with the microbes into the solid medium. On the right hand side, you are seeing the streak culture, where the bacteria are growing in the form of streaks. Thank you for watching my videos. Please like, share and subscribe. We'll meet again in the next video with some other topic. Thank you very much.